All right, there we are. So, again, good morning. Just good morning. My title was already announced very well. I wonder if you can actually hear me if I'm standing over here. Yeah? Is that good? Can I walk around? Do I have your permission to walk around? Excellent. I like to walk around. So. This is what I've got scheduled for you today to talk about. I wanted to talk very briefly about human cortical architecture and multiscale connectivity. Briefly because I think you've heard multiple speakers' perspective on this, but I'd like to give you mine in the context of what I'm going to present, which is, as the title already predicted, a lot of post-mortem ex vivo MRI and also post-mortem ex vivo optical clearing and light sheet microscopy. So there's a lot of anatomy, but what I hope to be doing in presenting the methods that we've been working on over the last 2 to 3 years is our perspective on using these methods to bridge what Rainer just told you about, which is in vivo human imaging of structure and especially function, and the real microscale anatomy that you've learned a lot about in the previous days. And in order to do that, we have to study the human cortex. We have to study its anatomy over multiple scales and down to very high resolution, but we also need to have a perspective to bridge it back to in vivo. And the perspective that we have in our lab is to do it ex vivo with MRI at much higher resolution than you could achieve in vivo, and I'll show you a lot of images about that. But at least it happens with the machines, the 7T and the 9.4, that can also be applied in vivo, so even though it will be maybe an order of magnitude less resolution, we learn a lot about what we could potentially see in vivo. Simultaneously, in those ex vivo samples, human brain samples that we use, we try to make them the whole human brain, or large parts of the human brain, not just tiny sections. And to also map those with light microscopy techniques that are capable of covering, in principle, those entire large pieces of human brain, so that we can cross-correlate between MRI and light sheet microscopy. All right, so brief introduction to human cortical architecture, or at least reintroduction into nomenclature as I use it. If you look at the cortical sheet, and I have to apologize because every time I run this presentation on my Mac, this little column here gets displaced. So imagine it to be right here on top of the cortex, right? If we look at the human gray matter or the cortical sheet, we've got an immense expanse that is almost everywhere 2 to 4 millimeters thick. And inside that expanse, you've already learned a lot about the cortical architecture that it has. And you've learned probably that we have myeloarchitecture there. So that means that there's variation in both tangential and also radial and tangential, so horizontal and vertical myelinated axons that vary over different layers in the human cortex. And there is, of course, cytoarchitecture, where we have differences in the morphology and the density and the size of cells. And of course, the most prominent one of those are the large pyramidal cells that we tend to have in layer 3 and layer 5. So that together, defines the prototypical 6 layers of the neocortex, both in myeloarchitecture and in cytoarchitecture, right? So just a reminder to you that both in myeloarchitecture and cytoarchitecture, we talk about the same layer 3 and the same layer 5 and the same layer 6. We tend to write it down in cytoarchitecture with a Roman numeral, and then in myeloarchitecture with an Arabic numeral, but it's still the same layer 3. The thing that changes a little bit now and then is that there are different sublayers. For instance, layer 1 can have quite a number of sublayers in myeloarchitecture, but not in cytoarchitecture. But still, it's the same layer. The one thing that I would like to add is that, of course, all of these layers are connected to each other, but also some of them, or even many of them, are connected to remote other sites of the gray matter, and we could call that the connect architecture. So that is the large range, long connections of certain neurons in some of these layers through white matter to other sites that can go very far in the human brain. Right? These can travel 10 centimeter or even 15 centimeter to another part of the human brain. That also brings the challenge of trying to image all of the architecture of the human neocortex is that we could zoom in with light microscopes all the way here to this and see single cells, but that usually confines our field of view to looking at this one column of cortex, whereas each of these pyramidal cells could have axons that travel all the way to the other side of the cortex, and they could also ramify along the way and have a very large axonal ramifications that span over many tens of centimeters or cubic centimeters. So that puts up the challenge that we have if we're trying to image this. So let's look a little bit at how to image this, and in doing that I'm presenting yet another introduction, a version of what you've seen before, which is multiscale architecture of the human brain, or in my case I'm calling it multiscale connectivity. So we can go all the way from the macroscale via the meso and the microscale to the nanoscale, all the way from the whole human brain down to where we could be talking about neurotransmitters and receptors. And along the way, if we look at the connectivity, we pass interesting connectivity phenomena, such as long-range association projections and short-range association projections, large bundles of axons that travel through white matter, and those would be accessible even with in vivo diffusion-weighted MRI. And that's a lot of what human macroscale connectomics, such as being done not in the HBP but in the HCP, you might be familiar with that project as well, the Human Connectome project. That main project that was driven by the NIH in the United States has actually just ended, and it's been followed up by the aging connectome and the baby connectome and the developing connectome. But the idea has always been to in vivo get as high resolution diffusion MRI as you could get in order to do what is called tractography. If you're not familiar with that term, I'll define it a bit better later. But it is a way to define and get a handle on those long-range white matter projections and map them out over the human cortex. So that you have access to with human diffusion-weighted MRI. But human diffusion-weighted MRI stops at about 1 millimeter isotropic. And from there on out, if you wanted to go to, for instance, the topography of the projection organization, like for instance, we could imagine that the projections of V1 to V2, Rainer has explained in detail how that looks on the cortex. Of course, a lot of information streams from V1 to V2. And we also know that a lot of information will be topographically preserved. So a lot from the center of the visual field in V1 will go to the center of the visual field in V2, and the same for the periphery. That means that there's a lot of axons traveling from V1 to V2, and they're topographically organized. But they are organized, topographically at a finer scale than a large bundle that travels from one part of the brain to the other part of the brain. So if you wanted to look at that, you would need higher resolution, and that tends to be beyond what we can currently do with in vivo diffusion-weighted MRI. Ex vivo diffusion-weighted MRI, what I will focus on a lot today in the first part of the talk, however, might have access to it. I don't have concrete examples of that to show you. I'm just saying that this is one connectivity phenomenon at the mesoscale that requires higher resolution and that justifies trying to get much higher than 1 millimeter isotropic higher resolution, and first to do that ex vivo, then to see perhaps whether it's translatable to in vivo. After that, of course, we can go into the cortex and we can get to these layered intracortical circuits that are actually made up by the cytoarchitecture and the myeloarchitecture. So those would be the circuits created locally by connections intracortically, not traveling through white matter within one layer, but also especially from one layer to another. And there are a lot of those connections. In fact, we know already that the amount of connections that any given pyramidal neurons make, so any synapses, you could say, that are dedicated on intracortical columnar circuit connections versus remote connections, both coming in and coming out, about 90% of them at least will be inside the cortex and will be over a span of 100 to 200 micrometers. That's where any pyramidal neuron spends, 90% of its inputs and its outputs, and only very few percent comes in remotely and goes away very far. Like, for instance, one number that is often called, that has been verified in the cat, is that in V1, right, where input is hugely important, obviously, right, and it has a huge layer 4 dedicated to it, you may have seen in the past days, only about 4% of the synapses of layer 4 neurons are actually dedicated to thalamic input. That's, mesmerizingly little, actually. So a lot of all of those many synapses, of those many billions of neurons, takes place in that intracortical circuit. And so to be able to look at that is another justification of trying to upgrade our resolution to the mesoscale, already also at the structural level. That, though, is at the border of what perhaps even ex vivo diffusion MRI could do, and therefore we then get into the realm of ex vivo light microscopy. Now, light microscopy in itself is a huge field and has many different modalities, right? There is a bright field or wide field microscopy that you can do in sections that you have seen a lot about. There's PLI, but what I will focus on today is a set of new techniques in the second part of my talk, which are optical clearing, making large, thick pieces of human cortex transparent, and then imaging it with a so-called light sheet microscope, which has the resolution of light microscopy, so 1 micron isotropic and beyond, real microscale resolution, not quite nanoscale perhaps, but certainly microscale, but it can do this at a very high throughput and at a very large field of view, and in that way it can be nicely overlapped with ex vivo MRI. Alright, and as soon, of course, as we go down even further and we travel down to single axons in synaptic contexts, we seem to be entering the realm of ex vivo electron microscopy. So if you really wanted to see a synaptic contact, right, a dendritic spine contact to an axonal bouton, probably we would have to go to an electron microscope, although I have to note that there is very interesting developments very recently in so-called super resolution light microscopy, where the diffraction limit, the limit of about 300 to 400 nanometers, 0.3, 0.4 micrometers, can now be broken on a regular basis with light microscopes, and they can image down to 100 nanometers. Getting closer, but not quite equal to EM resolution. All right, I will, of course, today focus on these two techniques and basically focus on these phenomena here, mostly on the mesoscale. If you have any questions, by the way, just raise your hand and we can do them while I'm talking. We don't have to save them for the end. So MRI then, MRI resolution. I can go over this rather quickly because Reiner's touched upon it, but I just want to drive home the point that he also made, that with the rather standard 1 to 2 millimeter isotropic resolution that we have at 3 Tesla for things like fMRI, but also diffusion MRI, because it's all kind of based on the same readout technique. It's all based on EPI, on echoplanar imaging, and that just at 3T it stops at about here. If you put that right next to, you know, that intricate architecture of the cortex over 2 to 4 millimeters, right, it really drives home the point how miserable that resolution is to have any chance of looking at layers. One would really have to get down to a much better resolution, starting at about 1 millimeter, preferably 0.8, right? 0.8 would give you a chance to maybe look at infragranular, granular, although a bit more as well, because the granular layer is quite thin, and supragranular. But it's the first point where it gets interesting, in order to maybe look at top-down versus bottom-up. And it's only when you get to, let's say, 1/3 of a millimeter on the side of a voxel, so 330 micron, that you start to get the resolution to distinguish the great majority of layers and some of the sublayers of human cortex. But realize, and again, same point that Ryder made earlier, if you want to go even from here, which is the best resolution we can do, not quite functionally, but at least anatomically, right? At 3T, and you want to go to here, you have 27 times better resolution, right? 3 times on a side, but it's in 3D, so we cube 3 and we get to 27. 27 times better resolution means we need 27 times better signal-to-noise ratio in order to get good image quality. Now, in order to get that, either you scan the square of 27 longer, the square of 27, I don't know what number that is, about 800, you scan 800 times longer, or you do something, you know, really transformative in order to get in roughly the same time, having a person in the scanner for about an hour, right, to get that amount of resolution. And this is exactly, that drives, you know, home the point of why we spent a lot of money on 7 Tesla and 9.4 Tesla magnets, because these already make the equation so much better. Right away, of course, things that have their SNR, signal-to-noise ratio, again, rise linearly with the field, right? They will have twice the field, more than twice the field at more than twice the SNR at 7 Tesla, more than 3 times the SNR at 9.4 Tesla, and so that takes a lot away of that, you know, nasty 27 factor. Not everything, but there's more tricks that we can play. One is a nice trick of nature. Reiner mentioned a couple of nice tricks of nature, such as, you know, hemoglobin kind of helping us to make fMRI contrast possible at all. Another nice thing is that T2 star contrast. which is the technical name, if you don't know about it, for the exact contrast that we look at when we look at fMRI, that is T2 star contrast. That's what the deoxyhemoglobin does, you know, it changes the T2 star contrast, but also some of the anatomical contrast that I'll look at. I'll talk about T2 star a lot. That contrast actually goes up more than quadratic, more than linearly, closer to quadratic with the field. So that means we get much more than 2 improvement at 70 and much more than 3 improvement at 9.4. And of course now we get towards, you know, getting to that factor 27. Another trick that we have to play is to build high-end radio frequency coils with a lot of receiver elements very close to the sample, because that also improves our SNR immensely. So the simple theory here is that if you get more smaller loops closer to your sample, closer to the head in terms of fMRI, also closer to the post-mortem brain sample in ex vivo imaging that I'm going to talk about, you can get much better than linear improvements. You can also get quadratic improvements. So if we go from an 8-channel head coil to a 32-channel head coil to a 64-channel head coils, we can get much better than linear improvements. And now the trick is that those improvements, more than 3 times from 9.4 and more than 2 times from building a better coil, they multiply. And if we multiply more than 2 by more than 3, we have much, much more than 6. And this is how, in a nutshell, I'm not going to drive the argument much further, this is how we get to the factor 27. By playing improvements on main field strengths, on gradients, a lot of efforts on specialized RF coils, which gets us then from the 1 or 2 millimeter down to the 330 micron isotropic. Now this is a post-mortem whole brain sample. I'll talk a lot more about this in the next 10 minutes of the talk, where indeed we get to the 330 micron isotropic, so that you can see that that is possible when we work with that specialized hardware. Any questions about that before I continue? No? Okay. And we'll have a look at actually at both of these machines and at these coils when we go to the scanner later. So I get to tell a little bit more about the details then. Yes?
Could you say something about the limitations that come with these specialized hardware? Yeah,
good question. So the limitations that come with the specialized hardware, when they work, they're fantastic, right? So now the limitations. They don't work very quickly because basically you build them yourself. And so this is we built these coils in-house. And so I'm already running a little bit ahead to my next slide because part of my answer to you was going to be my next slide, but that's well timed. We spent 2 years on conceptualizing, building, and especially also optimizing, debugging, and perfecting this coil. Right? So if you're serious about whole human brain imaging post-mortem, you don't use a human in vivo coil that has a hole at the neck. It has to, because you know our heads are still attached to our bodies very trivially, but you know that's where there are no coils, and they have to be further away from the brain because there's our skull, right? And there have to be some cushioning and some measure for having different brain sizes. We got a lot more signal, but by building this specialized container here that exactly can contain human brain samples, get all loops to all size very close to it, and get much more signal from it. But you know, when you build this coil really to a level where it can get very high SNR, you're also pushing your preamps much harder. They will break. They have to be fixed every 2 months or so. The coil has to be perfectly tuned. So you need manpower. You need serious manpower just to keep RF coils running. Another downside that you might mention is that it's not so much a downside as it is a consideration that you have to take into account that once you get this kind of data, it's not that you can say, oh, now my job is done. I plug it into my standard pipelines, right? My Brain Voyager, my FSL, or my FreeSurfer, and now I will immediately get my benefits. No, then you work very hard on changing all of your analysis pipelines. For one thing, to even load these images, because they're not used to that. The first thing that will tell you is memory error. We can't load an image that is 4 gigabytes, right? So you have to work very hard at every step of your analysis to make it work as well.
Yes. So actually this image on the left is after tuned preprocessing?
I didn't quite hear your question.
This image on the left is like raw data, or
it's,
is
yeah.
after preprocessing? Because actually we have some noise components and everything that you have to, like in free Tesla scanners, you have totally different filters to filter structural data and diffusion data. So actually here, about noise distribution and everything is that it changes. Like if you have parallel MRI acquisition, it's give you totally different noise distribution.
Yeah, good question. That's both a consideration and not. Where it's not, I can start with, we don't do a lot of parallel imaging in post-mortem. Because parallel imaging, as you know, if you do, I don't know, a multi-band or grappa, you cut your measurement time in half, you also cut your SNR in half. And I just told you how badly we need SNR, so now we're not going to sacrifice it again to do parallel imaging. Not in most of the cases of what we do. So this is unaccelerated imaging, which makes the whole noise story, and you're completely correct, that gets complex when you do parallel imaging. That makes the noise story easier. On the other hand, though, since we are squeezing for every ounce of SNR, even when you do standard, non-accelerated, 3 times Fourier transform encoded fMRI, when you look at your image reconstruction, we don't do what the scanner always does. Like, for instance, all of these coils, they do one separate image. One image looks like it was taken with a lamp on this side of the brain. One image looks like it's taken with a lamp. You have to combine them. If you do standard combination, you lose a lot of your SNR. You have to actually do complex combinations like Römer combinations, or at least covariance-weighted noise combinations. So we do all of those, and we do all of those offline. So basically, we get, when we measure for a weekend on a whole human brain, we get 4 gigabytes of raw data, and that goes into MATLAB, and it processes for 24 more hours. To get 4 gigabytes of very high resolution images.
So actually in this case you need to invent your own reconstruction algorithm in this case.
That's right. Yeah. So the scanner, this was the last image that the scanner could reconstruct. Everything I'm going to show you next, because this was the work we did 3 years ago, and we're going to go much further down in recon. The scanner can't even handle anymore. So the first thing we do when we walk on a Friday afternoon, we're going to measure for a weekend, is turn the scanner, well, we turn the scanner recon on, and we look at the quality of the basic 500 micron images to see, okay, we've got our transmit right, we've got our receive right. We turn recon off, and the scanner runs for 60, 70, 80 hours, and we don't see data. All the raw data goes to another server for the recon. Yeah. All right. So let's get to whole human brain high-resolution MRI. And in principle, of course, you could call this macroscale because it's whole brain, right? I had the whole brain in the macroscale region. But really what's been our mission, you can see from what I've told before, is to take macroscale field of view, but mesoscale resolution. So to keep the field of view to the whole human brain and see how far down we can get with MRI resolution by playing all the tricks that I said. So to summarize the tricks that I said, we go for 9.4 Tesla for reasons of getting that field effect. So that's the system you're going to see later. And 2 technical aspects of it that I'll tell you a little bit more about when we're at the scanner is that it has a parallel transmit system. In fact, the 9.4 Tesla has a 16-channel parallel transmit system, of which for the whole brain we use 8 channels. Parallel transmit is there to make sure that we actually get a decent image of the whole brain, because the short story of the downside of 9.4 Tesla imaging, or even 7 Tesla, is that when you use a single transmit coil to flip all the spins, right, and then you read out again, you'll get some dropouts in some parts of the brain. And simply said, that's because the wavelength of the RF energy got so close to the side of the head that you get destructive interference of waves inside the head. So you get bright spots somewhere and dark spots somewhere else. So great, you've got great signal somewhere in the brain, but it disappears somewhere else in the brain. So you need to send in 8 different signals with 8 different transmit coils and adjust their phases in such a way that they all constructively interfere everywhere and that you get nice signal. That's the short story. So you have to, that's another you could call downside or complexity. You have to do this if you want signal from the whole brain in at 9.4. So when you see me mention PTX, a lot of work has gone into shaping the RF pulse in such a way that you actually get nice signal to begin with. And then also we've got quite a good gradient system in the scanner, which helps especially a lot for diffusion, which I'm going to talk about a little bit later. And then of course, in order to get that high resolution imaging of the whole human post-mortem brain, we want very high SNR, and we want that very time efficient, because basically, you know, the more SNR we can get from 80 hours of scanning in a weekend, the higher resolution we can push. So also the aspect that I won't mention so much, but that also gets a lot of work, are the pulse sequences. So the real sequences that we run on ex vivo brains are totally different from the ones that you run in vivo. Because the ones in vivo, actually, frankly, they just don't even work. You have to go to a very different way of scanning. For one thing, you have a very low duty cycle when the post-mortem scans that we do run. You'll hear tick, 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 tick. And so it runs with a very small readout every time, where it gets a little part of the data every time. And that's basically because if you try to do a really long readout, like an EPI readout, where you hear this entire beep, right? When you hear fMRI running, you hear beep, 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 beep. And each one of such beeps is 40 to 50 milliseconds long, and it's the scanner reading out the entire case space, or your entire slice image. That only works because the signal in the in vivo brain is there long enough to be read out for 40 to 50 milliseconds. Ex vivo, you can only read out signal for maybe 10 to 15 milliseconds, so you have to make really short readouts. That's already one thing that you have to do. But all right, we've taken care of all of that. And so the first image that we did about 2 years ago, Rainer also showed you, was to go down to 200 micron. This is an entire human ex vivo brain. Here you see posterior, anterior. You see that it's about at the level of the superior cerebellum, and you see the hippocampus over there. And the thing you see is that there's very, very nice white gray matter contrast, even though the contrast is reversed, right? So white matter is very dark here, and gray matter is quite light here. And that is actually one of the things that happens ex vivo when you fix a brain, and all of these brains are entirely formalin fixed, as also, by the way, in most of the Jülich pipelines, except for the PLI pipeline, I believe. All of these brains are fixed, and what happens is that some contrasts reverse. This is a T2 star contrast. A lot of what I'll show you is a T2 star contrast. But you do see that there's a very nice contrast between white and gray matter. There's even some contrast inside the white matter, where you can see that certain white matter tracts that are more or less myelinated stand out inside the white matter, which is also interesting. And then if we magnify a little bit, you can already see that we, at this resolution of 200 microns, we can start to see some of the layered structure of the hippocampus. We didn't stop there. We pushed that another factor of 8, right? 200 to 100, again making that same point. Sounds like a little step, but it's actually a huge step, because it's 8 times higher resolution, 8 times lower SNR, at least 4 times longer running time to even get the whole brain encoded. But we are also capable of getting good SNR over the entire brain at 100 micron. This particular set of samples was actually a collaboration with Jülich, with Professor Zillis. You can see that this is actually 2 separate hemispheres, bisected here. So a left control hemisphere and a right advanced stage Alzheimer hemisphere. Where if I zoom it a little bit into the medial side here, once again you can see we have quite beautiful hippocampal layering in the healthy hemisphere. There's also quite some beautiful contrast in the subcortical midbrain structures. And you can basically, at 100 micron, you have the resolution to see some of the admittedly very, very extensive damage that was going on in this brain in the Alzheimer hemisphere. Then finally, the furthest we were able to push the resolution on the whole human brain was down to 75 micron isotropic. And once again, that's another factor of 2 above 100 micron, so a factor of 16 above 200 micron. And as you can see, we were capable of getting nice T2 star contrast over the entire brain. You've got your transverse or axial section, your coronal section, your mid-sagittal one. And of course, if I zoom in a little bit, here in the transverse one, you see that the contrast and the contrast to noise is sufficient to see a lot of detail in the midbrain, a lot of the nuclei there, the caudate, the putamen, the thalamus. Many of those are very well defined by contrast of myelin and especially also iron content, which T2 star is very sensitive to. But most interesting to us, to our lab, is to go and look in the cortex. And here you see an inverted, I apologize, this is superior, this would be inferior, piece of the motor and premotor cortex, where I hope you can distinguish at the projection here, I think it's visual, that inside the cortex at 75 micron, you can start to see significant layering here. And so although we are now going to go into a project, and this was again a brain coming from Jülich in collaboration with Katrin Amunds and Carl Zillis, we're going to verify this and investigate this more in detail, but this strongly suggests here that we have the resolution and the contrast to see myeloarchitectonics, and this looks suspiciously a lot like the inner and the outer layer of Baillargeau, kind of very significant hallmarks of myeloarchitectonics. They were actually labeled earlier with BI and BO. You may have seen that. That picture is going to come by again, and then I'll show you. So in principle, we have the potential here, you could say for the first time, to look at myeloarchitecture over the entire human cortex, and it's intact as well. right? You might have heard in the past days, I cannot imagine that Carl or Katrin or Svenja has not told you this, that the Vogts did a lot of work on myeloarchitecture, but they were so incredibly thorough that they actually never got to, you know, doing the entire myeloarchitecture of the entire cortex, right? So a lot is still a lot has happened since. Yeah, the myeloarchitecture has perhaps been underappreciated for the last 100 years, but it's also not seen no work. But still, there's a lot not understood yet. And here you have a data set that has the contrast and also the field of view and the integrity. This is not 5,000 sections, but it's an intact 3D data set that you can slice in any oblique way that you can to investigate myeloarchitecture. So that has one of our interests on working on in the coming while. Yes?
So regarding this like whole brain myeloarchitecture studies, it seems like inevitably you have some spatial bias over the whole field of view, right? Either like
Why so?
It seems there that I see some drops in the signal. More
Yeah. So
posterior,
it's
right?
darker here and it's brighter here, right?
Right.
I'm gonna get to that. I promise I'll get to that. If after I've talked about quantitative T2 star, QT2 star, you still have questions, then just ask the question again. You're fully right. It's so important that we work on this. Yeah. All right. Continuing. Because of course, although this says a lot about myeloarchitecture and something about intracortical circuits, it is very indirect, right? It still doesn't have a shot to look at connect architecture, and it still doesn't talk directly about connections. And of course, the technique in MRI to look at connections with is diffusion MRI. And I imagine that Svenja may have told you something about diffusion MRI. Yeah, so again, I'm taking the liberty to give you my brief one-slide recap about what diffusion MRI is, because I do assume that you know something about it already. In diffusion MRI, we look at nothing else than the diffusion of water. The passive motion of water, the thermal motion of water, Brownian motion, right, as it's known. The fact that, you know, if water is not at, basically, you know, if it's not ice, the water molecules will randomly walk around. And when they do so, they will bounce into things. And they bounce into things in a very structured way, especially in white matter, or when you have a enough resolution, also in gray matter once you get into one of those very well-defined myeloarchitectonic bands, right? Remember the radial and the tangential ones. So for instance, if we have an organized bundle of axons over here and a water molecule is, you know, bouncing around there, it will, on average, over time, displace further in the direction of the axons because it has less barriers, less hindrance, less restriction in that direction, than it will perpendicular to the axons because it will bounce into the borders more. So another way of saying that is that the parallel diffusivity, d parallel here, will be larger than the perpendicular diffusivity, d perpendicular here, which is exactly what we call anisotropic diffusion. So anisotropic diffusion is the hallmark of structured tissue that gives us, also with diffusion, a way of looking at the microstructure of tissue. And micro is one important term here, because the interesting thing is, of course, that this distance here might be 1 to 10 micron. So although with diffusion MRI you have signal on the level of, you know, hundreds or millimeters of voxels, what that signal is sensitive to is the microstructure of tissue, and that makes it interesting. So these are actually what post-mortem or ex vivo diffusion MRI images look like. What counts is how much diffusion weighting you put in there. And for instance, that's quantified by the so-called B value. And B value is a bit mystic when you first get into diffusion MRI, but it quantifies how much we weight the signal for the amount of diffusion that goes into a certain direction. So every single image is sensitive to diffusion in a certain direction and less into others, and it has a certain sensitivity. For instance, here it has very little sensitivity, B of about 200, 2000. At B 4000 ex vivo you start to see some nice contrast, and here's at B 8000. And you can see that you get contrast at the higher B values in white matter, right? Which is not there in standard anatomical imaging. And the precise contrast that you get is that the places in white matter that are perpendicular to the diffusion direction that I chose to measure, because every single image I have to choose one diffusion direction, right? And I will get many images with many different diffusion directions. But the places which are perpendicular to the direction that I chose will be bright, and the ones that are parallel to it will be dark. right? And so if I get many of them, I can kind of model my way back to what the direction must have been, because it will be the direction, you know, that is very dark in a direction that when I measure in that direction, and very light when I measure perpendicular to it. That's all. That's what diffusion imaging is about. Yes.
How much are you restricted to very freshly fixated tissue? So could you also do that with long-term fixated?
Yeah. All of what you see here has been a long-term fix, and in fact, I would argue that any whole human brain is long-term fixed because it takes months at least to fix it all the way through, right? But we've done.
Okay.
But we've done this from brains that were fixed from about 6 months on to brains that have been fixed for 3 to 4 years.
Okay.
Yeah. What you see is post-mortem interval counts much more. So fixating the brain quickly after death is always the most important thing. That will create differences between brains in the quality of in the amount of diffusion that is still left. That can range from can range across 300 to 400%. So we have really bad brains and really good brains. Post-mortem interval, it changes, you know, that quality 10 to 20% over the first year, and then it stays constant for very long. So that doesn't matter so much how long we have it in formula. That was another question that came up there.
How does the preparation steps of ex vivo brains affect diffusion measurements? And pretty much, yeah.
Yeah. The short answer is very badly. So anatomical T2 star weighted imaging on postmortem brains, if you work for 2 or 3 years on the coils and the sequences and the 9.4 scanners, can be very thankful because you can get the 75 micron isotropic, right? Diffusion is much harder. Diffusion to start with is much more SNR starved than anatomical imaging. So anytime you see, I don't know, 1 millimeter isotropic in vivo now, right? MP-RAGE, or, you know, SPGR, as GE would call that, you will see at least 8 times that resolution on diffusion. So diffusion will probably be 2 millimeters. And that's because diffusion has much less SNR than many other measurements. The short reason for that is that what I just explained, right? Excuse me. I just explained that parallel perpendicular. What actually happens with diffusion is if the fiber direction is parallel to the current diffusion gradients, its signal gets pushed down. If the fiber is the same, is perpendicular to the diffusion gradient, the signal stays the same. So diffusion pushes signals down, so you have less signal left. And so it's already very difficult. Now to get to your question, it's already difficult in vivo. It's even more difficult ex vivo because diffusion ex vivo is only about a half to a quarter of what it was in vivo. And most of that is due to temperature drop and fixation, right? So fixation decreases the amount of diffusion that's left a lot. And you can see the struggle that we have with fixed, right? The question was asked. You can see the struggle that we have with fixation in diffusion because what we generally do when we work with slightly smaller samples than a whole human brain, we will fix them. We will have to because we want to scan them for a very long time. They will not stay fresh for very long. We will fix them, but once they're well fixed, we will generally soak them in PBS with some azide, you know, to keep the fungus out for weeks to make sure they get rehydrated, which gets our diffusion back from a quarter of the in vivo value to half of the in vivo value. And otherwise we get, you know, 100% more signal again. However, on the whole human brains, we don't do this for 2 reasons. One is, it was already difficult to fix them. It is just as challenging, or perhaps even more challenging, to then assume that you can wash out all of the formalin down to the middle of the brain, right, and have it completely rehydrated again. And second, and very importantly, the anatomists that you tend to work with tend to say, oh no, this brain is staying in formalin. You know, this is my precious sample. You know, I'm going to dedicate 20,000 man-hours to this. You are not taking this out of formalin because, you know, after that, after you've done your scanning, which is all good and fine, I want to do my sections and I want to do my PLI or my cytostains or my whatever. And so on whole human brains, we have to live with that quarter of signal, which is really bad. I will get to occipital lobes later, where we can play the game a little bit differently. Yeah. Yes.
So what is the maximal B value that you can obtain?
So this is a pure trade-off. Higher B value costs SNR. Higher resolution costs SNR, right? If you measure longer, you win SNR again. So it all depends on the resolution that you want.
So the stabilization of the whole system also has to be
Exactly. Yeah. So the game we have to play ex vivo is, this was B8000, but we did it at 1 millimeter isotropic, and we want to push much further. I'm going to show you B values and resolution, and you're going to get a sense about this. But indeed, for ex vivo imaging, it's always a measure of, okay, we run for very long, but it tends to also only be 80 hours or so, and then it has to be done. But if we go for really high resolution, we have to play a little bit less B, otherwise we don't get SNR anymore, we can't analyze the images, but we'll get less diffusion contrast. So this...
So if you even sensitize the cells to like the higher B value, you will drop the resolution. So if you use this epi acquisition, you cannot disentangle whether this was like isotropic or crossing angles.
Oh, we can in the end. You're right to say that it's a difficult trade-off, whether we go to high resolution or high B values or somewhere in the middle of both.
Depends on the algorithm later.
Yes, but also in order to look at crossing fibers, what you need is a high enough B value and enough directions.
That's true.
Right. And so 8,000, you can see that 2,000 is quite low ex vivo, even though in vivo it's quite nice, right? 4,000, 3 to 4,000, you start to get nice contrast. 8,000, fantastic contrast, but maybe a bit too much, and you have no chance in getting to the high mesoscale resolutions that we want. So you'll see that, you know, the short answer to your question is we tend to push B values between 2 to 4,000, right? Where 4,000 we run that at slightly lower resolutions, and closer to 2,000 we run it at much higher resolutions. Yeah. All right. So for instance, here's some work that we did on a so-called steady state free precession sequence, which is one of the different sequences that you don't tend to use in vivo. And we got down to 450 micron isotropic. So that is more than 8 times, let's say 10 times the best that can be done in vivo. Here actually you see, right, quite directly what we're talking about here. At that very high resolution, low B values look brilliant. Middle B values look good enough, right? But once you start pushing to the B4000, you know, the SNR of this image is, it's at the border. One upside that you have with diffusion is that you don't actually have to work with the SNR of this image alone, because of course with diffusion you need to get at least 30, preferably 60 directions. So virtually in your modeling you're averaging over 30 to 60, and so, you know, the SNR in your data set in diffusion is a lot better than the SNR in one single image. But you know, it does degrade, and you know, you have to stop somewhere, because you can get at the point where there's too much noise in your image and you can't analyze it anymore. Pushing that a little bit further still, and the best images that we've currently got, we got down to 400 micron, which is about 16 times better than 1 micron isotropic, the best that we can do in vivo. And I hope you can appreciate that even when you have single images here, this is the low B value image, this is the high B value image. In the low B value, you see you have quite exquisite anatomical contrast. In fact, you know, 400 micron is something that we cannot really achieve in vivo, even for anatomical imaging. And now here we're doing diffusion imaging at that resolution, and you can see that indeed in the low diffusion weighted or zero weighted imaging, you have the contrast, or even better than the resolution and the contrast that you're used to in vivo anatomy. And we can now do diffusion with this, right? This is the diffusion weighted data. A single volume will look very noisy, but of course analysis goes over many such volumes, and here you see some analysis results, and you can see that simple analyses like DTI, or in this particular case ball and stick, will show you, and this here is a stick fraction map, so how much of the diffusion in a certain voxel can be explained by anisotropic diffusion, different in some directions than others, rather than isotropic diffusion, the same in all directions. Shows you beautiful, almost anatomical looking white-gray matter contrast, and also it already starts to show you higher density fibers, such as in the corpus callosum, the splenium in this case, but also the genu, and the corticospinal tract versus other places in white matter. If you look at direction and coded maps, so of course we don't just have the anisotropy, but we also have the clear direction of anisotropy. And here you have the standard coloring, which means anterior to posterior is green, which is why the cingulum over there is green. Left to right is red, which is why the corpus callosum is mostly red. And then up down, inferior superior is blue. So like for instance here some part of the midbrain part of the corticospinal tract will be blue. We have that contrast over the entire brain, and where you could get this image in vivo from a 2 millimeter isotropic one, once you start to zoom in is where you see the value that you get. Where normally the corpus callosum and the cingulum are 1 or 2 voxels, now we have many voxels across those. But of course it gets even more interesting once we start to look not just on these very large tracts that we can also delineate in vivo with 1 to 2 millimeter isotropic, but once, such as again here the corpus callosum, this is a mid-coronal slice through the brain, right? And here you have the corpus callosum you're looking onto. Here's the cingulum, which you're now looking straight onto. It will be going through plane in green again. Here's the corticospinal tract. But besides being able to follow those tracts, you can see that I didn't quite line out white and gray matter here, but here at the edges, you're going into the gray matter, and you can see you can follow the directions into gray matter. So you now get gray matter contrast as well, and you can see that dominant radial diffusion that you also might remember out of the myeloarchitectonic picture that is in many parts of gray matter. You can start to see that. And so there is some hope of being able to track fibers with tractography techniques into gray matter rather than just stopping at the white gray matter boundary. Yes?
Could you even resolve that to layers, so to see where these radial...
Good
axons-
question.
Okay.
Let me answer your question after I've shown this. I mean, here you see, for instance, whole brain tractography done on that 400 micron data set. You can see that indeed we get a beautiful tractogram, perhaps slight, less quality here, but beautiful quality up here. You actually see, the gyrus is all mapped out by these fans of inserting fibers. You can see here we've zoomed in and put, the low B value weighted image below it, so that you can already see that you managed to track into the gray matter. And one bias that has been discovered in vivo macroscale diffusion MRI tractography based connectomics is that if you have low resolution, 2 millimeter, even 1 millimeter isotropic, what the tractography algorithm tends to do is it tends to follow them always into the gyral crowns, but it does not tend to connect the gyral walls anymore. Why? Because the tractography algorithm is a model, and the model thinks that traveling straight is better than taking sharp corners. And that is though what happens in gyral walls. In gyral walls, if this is the, in sulcal walls, sorry, if this is the gyral crown, the fibers will tend to insert straight here. They will bend, shallow here, but they'll start bending really sharply in getting into the sulcal walls. And so if you have poor resolution, that is what you'll think happens, that gyral crowns are connected a lot and sulcal walls are not. But already the resolution, even if you have such a naive tractography algorithm, helps a lot in, fanning out towards other parts of the cortex. To get to your question, and I haven't got that slide in today, so I'll just say it verbally. Can you see where it terminates into gray matter? No, I don't think so. And the reason is that diffusion MRI cannot distinguish between myelinated and unmyelinated. It cannot distinguish, for instance, between axons and dendrites. So if you think about the cytoarchitectonics of this column of cortex here, and now let's focus on the layer 3 pyramidal cells, you'll know that here's the pyramidal cell. Here are its axons, right? And here's its apical dendrite extending all the way up to layer 1. Those dendrites also tend to form dendritic bundles, and the axons tend to bundle up. Diffusion MRI can't see the difference between a dendritic bundle and an axonal bundle, right? So it will see radial diffusion all the way through cortex, and I'm about to show you a picture at even higher resolution on a smaller piece of tissue. But it's probably, the tractography algorithm is probably going to follow the axon, go straight through the cell body, follow the apical dendrite all the way to layer 1, and so it's going to make you think everything is connected to layer 1, which is not true. So honestly, I think there's a lot that diffusion MRI at high resolution can tell us about whereabouts in a tangential sense certain fibers insert. I don't think it can tell you in which layer it stops for that reason, because there's nothing in the diffusion signal that can distinguish between where the axon ends and where the dendrite begins.
It's still more bundled, I guess, no? Just radial axons, like in this myelo architecture, or?
I'll get to it a little bit as I get further on, but I think that I don't think there's hope for that, honestly. Was there another question there? Yeah.
I thought there was a way of looking at myelin in diffusion.
There is, yeah, there is. So what you can do, of course, is, and it gets more interesting, and it's the trick we want to play. So let's put this image on top of the 75 micron myeloarchitectonics, and now use our knowledge that these tracts are likely to end right in layer 3, which is just about at the height of the inner layer of Bayard-Gay, right? Yes, you can do that, and yes, we should do that. I'm just saying that the diffusion alone doesn't have that information. We will need to put it in either from assumptions or from the other modality. We have that other modality I showed you. So of course, in combining these data sets and doing clever assumption-based modeling, but valid assumptions, we can get quite a bit further. but the layer of termination will not be coming from the diffusion data.
I've actually used this bone and stick model on DTI, which is quite limited.
Yeah.
So why don't you trust these data-driven methods like ensemble average propagator just to
Oh sure,
data
yeah,
and
yeah.
calculate?
No, I don't here today have any modeling preferences. I have an interest here in generating ex vivo diffusion data that is suitable for all of those, right? So we do ball and stick, we do CSD, right, constrained spherical deconvolution, or propagators. And in fact, what you need for some of those is multi-shell data, multiple b-values.
Yes, yeah.
And I'm about to get there. Yeah, I'm about to get there. All right. Let's take resolution one step further, but let's also try to answer some of your questions where it comes to intracortical and multiple b-values. And for that, we go to intracortical diffusion imaging, so to get enough resolution to have contrast inside the cortex. Now 400 microns certainly is good there, but we thought if we limit ourselves to what will still be for microscopists huge pieces of tissue, but slightly smaller than the whole human brain, can we upgrade the resolution to the point where we can look at, you know, intracortical? This actually makes my point to you, right? So for instance, let's focus on layer 3 over here. Here are our pyramidal cells. Here are certainly where the axons stop. Here's where the dendrites begin, which are neither visible in Nissl stains nor in myeloid stains, but all of these dendritic bundles also go up. And so diffusion will come in here and it will see arrows all the way going up and it will only stop here, right? Here we have an early image that we did of a rather small piece of about 4 by 2 centimeters of human pre and primary motor cortex. This is actually the central sulcus. This is primary motor cortex over here. This is medial over here. And zooming in on this image over here, you see that at this resolution, 160 micron, diffusion weighted imaging, again, we have intracortical contrast that starts to look like we can distinguish the great majority of layers. And it has some semblance of that inner and outer layer of Bayer-Gay, which will have tangential rather than radial diffusion. So we know that if we look at diffusion, we should have radial diffusion probably all the way through, but tangential diffusion only at certain points. And that's what this image already starts to show. Here, just to show that if you do a simple DTI analysis at 330 micron isotropic resolution on about 60 directions, so once again we're coming to the number of directions. It was done with 60 directions. Indeed, if you have white matter, gray matter, you can see that radial diffusion all the way through cortex we can very easily get, and it's the main topic of intracortical diffusion MRI that we will also always get, right? So we can get the radial directions of axons and dendrites everywhere. But the challenge is actually to get to also these kind of crossing, you know, 90-degree crossing fibers, and it's actually what in the analysis, we're at the ends of some of our data acquisition now. In the analysis we would like to focus on. So we wanted to take this type of data to larger samples, not quite the whole human brain, but we wanted to stick close to the 200 to 400 micron resolution and go to, in this case, to entire occipital lobes. So we built another coil, once again, right? We have to build specialized hardware. If we want to push the limits, we have to decide on the size of the tissue that we want to focus on. And so in this case, it was about 8 centimeter cylinder that can house an occipital lobe. And with this specifically built 9.4 Tesla coil, we can get the 60 micron isotropic anatomical imaging, where once again, if we zoom into, this is the calcarine sulcus here, we can see beautiful layering going on there. But of course, you want to take that onwards to diffusion imaging. First though, here's the question that you had earlier about the biases. Because now we wanted to answer the question that it looks darker, lighter over here than it looks over here. And this is all down to the fact that this is closer to a coil, which lies here, and this is far away from a coil. And so quantitative imaging is the answer to that. Quantitative imaging, you get multiple images, all of which have the same coil profiles, namely bright over here, dark over there, at different echo times, and then you fit a model that fits an exponential decay model to all of this, which then puts all of the coil profiles, all of the bias fields in one image, which we call the S0 image, and it puts then the contrast, the T2 star contrast, in another image, which is entire bias field free. And that you can see does not have these biases anymore, but it does still have, and here you see a surface reconstruction of the occipital lobe sample, the calcarine sulcus. Here's a coronal cut through it. You can see here again is the calcarine sulcus. You can see this, the stria of Gennari in the quantitative T2 star, the QT2 star image, run all the way, and in fact, if you look very carefully, you can see it terminate just about here and just about here. So this is probably where V2 will start. And we can do that neuroanatomy in 3D in intact tissue samples. We also made the T2 star, the quantitative T2 star work in the entire brain. So here you have 100 micron T2 star weighted with bias fields and 200 micron quantitative T2 star without bias fields. Because obviously getting 4 volumes in order to fit 2 takes time again, right? And so once again there's this trade-off between, do you get 4 volumes at 200 micron and do quantitative, or do you get 1 volume at 100 micron and get non-quantitative? Here the resolution is better. Here the SNR and the bias fields are better. This was one kind of proof of principle where once again we look at the calcarine sources on the left side, and we look and we can beautifully define the stria of Gennari all the way through. In fact, in both images, and in fact here you start to be able to see 2 layers, right? You might in fact be able to see both layers of Bayard-Gay even in V1, and you might see hints of architecture changing over here. And the question of whether higher resolution non-quantitative versus lower resolution quantitative is better in terms of defining myeloarchitectonics, well, that's one of the focus of our work right now. You can see kind of the pros and cons of both of these images, like, you know, eyeballing it right now.
Have you combined any of these approaches with Nick Weisskopf's in vivo histology approaches for the multi-contrast imaging?
Yes, we have. And in fact, we also do Nick Weisskopf's contrast ex vivo. That focuses especially on, for instance, T1 contrast. In our experience, ex vivo T2 star contrast is easier to get to, better to model quantitatively than T1 contrast. So yes, as a short answer, yes we do, although we slightly prefer the T2 contrast. And not all of the in vivo modeling frameworks that Nick does also work ex vivo. Same story. Not everything ex vivo works that way. Yeah. I can see that my time is just about up. Now we could do 2 things. I could go to the rest of the talk quickly because I answered questions and I took my time, right? And we run a little bit late. Or do you want to stick to the time schedule? I'm also looking at you here. There is lunch. You should eat. But I'm kind of asking, like, do you mind if I keep taking my time to answer your questions and take it a little bit longer? Yeah? Is that good? Okay, then I keep doing that. Although I'll go a little bit faster. We're not supposed to go all the way through lunch, obviously. So here we get to your questions about multiple b-values. And so here's the occipital lobe with a STEAM sequence, which has a lot of benefits ex vivo. The KT is for parallel transmit, so we had to make a parallel transmit version of STEAM in order to make this work. And with that, we could get to 400 micron isotropic, same as the very, very, very best resolution that we could get for the whole brain. But here we have enough SNR to do that over a range of b-values. So here you see B3000, 4000, 5000, and even 7000. And so with this data set, right, we have at very high resolution also the high b-values to get the crossing fiber models, right, like we mentioned before. And also analyzing over the b-values, we can do some more complex modeling frameworks in diffusion MRI, such as NODI, such as the diffusion propagators, such as CHARMED, that allow you to look not only at the local direction, but it also, for instance, can tell you something about what is the fiber density there. Like, are there, in this voxel, which runs in this direction, and this voxel that runs in this direction, can the diffusion signal tell us about the fact that it's more likely that here we have, I don't know, 1,000 axons per square micron, right? Well, per square 10 microns probably. And here we have more like 500 of them. So we do have that full resolution b-value trade-off over here. Now we use that to do some basic modeling, and there we saw that already this is enough to really start to look at some of the mesoscale but long-range kind of probably topographically organized connectivity of the early visual system, right? So here we kind of connect to what Rainer was saying a lot. Rainer has been looking a lot on this part. Here's V1, here's probably V2, and again V2. But we know, of course, that somehow from LGM, you know, the fibers will travel into V1. So how did they do that? Well, this answer already kind of gives you that answer, right? Here, so we're looking at a coronal slice through one occipital lobe. Here is the lateral ventricle, right? And so the optical, here is V1, or the calcarine sulcus. So the optical tract will be coming from the thalamus somewhere over here, where my hand is now, and it will travel inwards, and it will travel it will curve around the lateral ventricle in order to get into V1. And so we could delineate all of that. So here is the optic tract, right? comes in like here, goes through there, and behind this it will curve into the calcarine sulcus. And I believe that, yeah, this will not show it much, although you do see it curve around. If you look through the lateral ventricle here, you'll see it curve around. And it does that, you know, in the vicinity of many other tracts that also travel there. Very busy highway system over there, right? Many bypasses. So it's interesting to see how things travel. Like here is one arm of the superior longitudinal fasciculus that, you know, that connects more superior parts of occipital and parietal cortex. And there's also the sagittal stratum, which also travels at a different part around the lateral ventricle. And in fact, if you look at the direction encoded DTI fit, diffusion model fit to white matter, you can actually very nicely distinguish the superior longitudinal fasciculus, the optical tract, the tapetum, the sagittal stratum, and with tractography reconstruct how those tracts are going to connect early visual areas. And so that's a matter of ongoing analysis right now that we have this data. The one thing that on the acquisition point that we want to do there is to see if we can sacrifice a little bit of B value, and it comes back to your question, so not quite to go up to B7000, but to stick at 4 to 6000, and see if we can get down to 200 to 300 micron resolution. Here's an example of a piece of a cat brain that we did with that, where it's easier to get to 250 micron diffusion resolution, where for instance you can see, once again, large white matter tracts, radial diffusion all over the cortex, but the new part that we didn't see before is that at this resolution you can also start to see the dominant tangential diffusion all the way up there in layer 1, because you may remember that in the myeloarchitecture, all the way up there is what's sometimes called the Exner stripe. There's lateral fibers at the surface of the cortex, and we can start to see that. Here's one of the very early images, and we can do better right now, where we can get 250 micron isotropic resolution diffusion weighted data of the human occipital lobe. And so we're seeing how far we can take that. I've discussed most of this, the outlook on MRI. I mean, trading off B values for resolution and getting either higher resolution or higher B values is a major thing. But what I wanted to spend the last 10 to 15 minutes on, and I'll go through it a bit quicker than through the rest, is to combine all of this with 3D histology. And we want to do this in its verge of optical clearing and light sheet microscopy. And in fact, one of the course participants, Sven over there, he's my own PhD student, and he's done most of this work. So that also connects you, not only to me later, I'm happy to talk to you, but also to Sven himself, that has done a lot of the clearing work that I'm about to show you. I'll take it a little bit faster, but I'll tell you that what we're focusing on for now is cytoarchitecture. And you've seen a lot about how cytoarchitecture can be done in the big brain, namely do many sections and then try to painstakingly 3D reconstruct that into the brain. Now, I don't mean to sound degrading of this. This has been a major data set, you know, that has changed how we can look at cytoarchitecture. But of course, it's very labor intensive, can only happen in 1 or 2 labs in the world. And to get this to the single cell level is, if not impossible, then extremely challenging. So there's been a lot of buzz in the recent years about optical clearing. In the mouse, that has now become a basic neuroscience technique. Entire mouse and rat brains can be made optically transparent and can be imaged for, in fact, any kind of genetic reporter, but also for cytoarchitecture, with light sheet microscopes, which are a very fast way of getting huge data set at micron resolution. Now the question we asked is, can we do this in the human brain? That's not as easy as it sounds. For one thing, the human brain is way larger than the mouse brain, as you very well know. But also, the adult human brain is much more myelinated. We don't have nice perfusion fixed mice like you tend to have, you know, in mice studies, but we're talking about long-term formalised fixed brains. And it's especially difficult in thick tissue, so like thicker than 1 to 2 millimeters, which is probably what you need if you want to look at the intact cortical sheet over long stretches of it. And therefore, it is also difficult for dense labels, and especially it's very difficult to scale. Like the best that Nick Weisskopf's lab, in fact, was able to do before we started this work was to look at 1 cube of 5 by 5 by 5 millimeters, and to stain that with antibodies and to image it took them 1.5 years. So the focus for us was to try and do a similar thing, but at 50 times a larger piece of tissue. And in fact, we chose that kind of 40 by 30 by 5 millimeter slab of tissue to be the exact piece of tissue that I've been focusing on a lot, which is a coronal slab through the calcarine sulcus, which contains V1 and V2. And of course we have to be extremely time efficient and cost efficient in order to make cytoarchitecture in at least that intact block work at close to 1 micron resolution. Now for that you have to clear your sample. So this is the sample when you start with it, and this is what the sample looks when Sven is done with it. And as you see, you can now look through it. And that is not only just a really neat trick, it's also completely necessary if you want to image optically through this piece of tissue, because in order to image the last millimeter of it, you will have to be able to send fluorescent, you have to be able to send light to it through all of the 4 millimeters above it. This is 5 millimeters thick, and it will have to also come out again through the side somewhere or through a lot of tissue in order to be detected again. So what Sven developed is what we call MASH, Multiscale Architectonic Staining of Human Cortex, which is a solvent-based clearing, which takes only 10 days for 5 millimeter thick samples for both clearing and labeling, and so then we can take it to the light sheet microscope and do 3D cytoarchitecture. And it's also something that is extremely cheap, so if you can imagine you had to do this with antibodies, it would be very, very, very expensive. And so you could maybe do it once, but to make a structured study out of this, or to even think about doing this to the entire occipital lobe, which is of course what we really want to do, would become very prohibitive. So Sven found some beautiful dyes, fluorescent dyes, which we call the MASH dyes, which are small molecules, so they're fast diffusing, in contrast to antibodies, which are very large molecules and slow diffusing. And in fact there are 4 different dyes, AO, NR, MB and MG, which you can use to color cytoarchitecture in different bands, which is extremely convenient, of course, to, for instance, both do nuclei and a Nissl-like stain, and perhaps then maybe in the top 100 micron another antibody that you like a lot, such as, I don't know, MBP or parvalbumin or neurofilament. And also going into the red into the far red allows you to image really deep, because of course all the signal loss in thick tissue, even when it's cleared, is due to scattering, and longer wavelengths, more red and far red, scatter less. Also, in order to make those dyes work, you need what are called better rims, refractive index matching solution, because the real transparency of the tissue only comes when you embed it into a fluid that is refractive index matched to the tissue itself. And so that is a very large and important part of the clearing. And we had to develop new rims in order to work together with the dyes. However, once we did that, and I'll start my movies and have them run while I tell you, once we did that, what we are able to do now is to take 2 to 5 millimeter thick pieces of cleared human tissue, and you see without sectioning, which brings all the pain of having tears and having wrinkles and distortions and needing to align this afterwards, we can do the kind of cytoarchitecture that you do in sections, but we can do it on the intact sections without any need to align afterwards. And so here you can see with the MASH AO, which is the green Nissl-like cell body labels, all the way through, this is now, you can't read this but I can, 3 millimeters in, through 3 millimeters worth of tissue, going all the way in, and you can see cytoarchitecture, I hope you can recognize all the way through. And of course then you can use that to do 3D reconstructions of your cells rather than have to work with the 2D data at the cellular level. MASH NR is a red cell body label, once again a small molecule marker, and red and far red, like I mentioned, and this is the illustration that we did of this, is extremely good to image very, very, very deep. And so the rest of what I will show you is mostly the red and the far red marker, the NR and the MB. And so here we have 5 millimeter thick tissue, and I'm showing you the plane at 500 micron, which has mostly layer 1 here, but here it starts. 2.5 millimeters in, and you see the cells over here, and 4.5 millimeters in. And in fact, we quantify the cell signal, the yellow signal here, and the background, which here we plot in blue. And you see that the signal to background stays very good and very well separated, all the way 4.5 to 5 millimeters in. And that's very important because with light sheet microscopy, where you send a sheet of light in from one side of the tissue, and then you detect on a CMOS camera on the other side of the tissue, and you detect an entire plane at the same time, and then you go deeper, entire plane at the same time, you have that loss, that scattering, both in the excitation, right? Signal drop goes in the excitation direction. Here, signal scatters more and more and more, and there's less excitation here. And you have it when the signal travels out in the emission, because it has to travel through more and more tissue here, so that signal will also drop in that direction. Now here is some example of the MASH MB stain, and this is a far red Nissl-like cell body label, and it's one of our favorites because with the far red we can image really deep. And I illustrate that both here in showing you a basic MASH MB stain. This is, I think, V2, and this would be layer 4, this would be layer 2 and 3, and here you see layer 1. And here you see the running stack of images that we inverted in intensity, right? So we made bright black. Why? Because then it looks like a classic Nissl stain. And if you've looked at classic Nissl stains, I hope you will recognize that this here looks very close to a Nissl stain that you would do on a 2D section, except now that we can do it on an intact piece of tissue. And you see layer 4 over here, the pyramidal cells in layer 3 over here, layer 1 over here, and you can even see, I hope you can appreciate, you can see the dendritic bundles, or what's sometimes called the microcolumns, right? The fact that these cells tend to be grouped into 20 to 50 to perhaps 100 micron thin microcolumns in layers 2 and 3. All of that cytoarchitecture is visible, and we can now analyze that in 3D. And so by now we're about 3 millimeters in, and this is quite an uncorrected image. The brightness starts to fall off a little bit, but we can still analyze all of this image. Now I'm going to skip a little bit some of the specificity and show you the very last part that I wanted to show you in the interest of time. So does this now allow us to do kind of the full multi-scale imaging, going from the macroscale to the mesoscale to the microscale, and connect the data that we did with MRI, right? Like for instance this occipital lobe data here, all the way to the signal that we might get from a single cell here in V1 or in V2. And so in order to do that, we did the MR imaging that we always do, so the quantitative T2 star imaging at 200 micron isotropic on an occipital lobe, and subsequently worked on 2 5 millimeter thick slabs that were large enough though, about 4 centimeters on each side, to contain V1 and V2 in them. And so on the front block of tissue over here, here you see it after clearing, with the combined MG nuclear stain and NR cell body stain, we can get, as you can see, entire fields of view that can cover multiple cortical areas, but we can also subsequently, with the light sheet microscope, zoom in enough in order to get single cell level signal that is sufficient for us to do the entire cytoarchitectonic classification of that tissue, exactly kind of correlated to where the MR signal would be. If you go to the other, the back tissue block, here again it is after clearing, and this was, where we focused a little bit on V2. We did a magnification where once again we have sufficient contrast in order to classify all cortical layers and even the sublayers. So putting this next to a classic cytoarchitectonic atlas where we're capable of recognizing all of these cells. And once again, of course, and I will show you the movie, this is in the context of something that is not just a 2D section, which it looks like when I show you the labeled one, but this is in the context of a deep stack that will run and will allow this all the way through, and it will allow this in 3D. And also, if we zoom in even more, and you've seen this image before, we can zoom in so far that in fact we have the 3D resolution to coarsely but truly segment pyramidal cell bodies in 3D. So we can really go from the brain all the way to the single cell, and so that's what we want to focus on in the lab, in combining these techniques in the same pieces of tissue. Very quickly, the outlook for this. So scaling this further is something that we really want to do. So we would like to scale the imaging of this further to something the size of an entire occipital lobe. But in fact, MASH is not the bottleneck there. We can clear and we can stain entire occipital lobes in 5 millimeter slabs, but the imaging becomes the bottleneck. And one of the things that becomes the bottleneck is that in classic light sheet microscope, the light has to travel through all of the tissue. And so we're moving now to so-called oblique or die spin geometries, where you rotate the whole microscope by 45 degrees, and your light sheet comes like this, and now your tissue can be arbitrarily wide, but still 5 millimeter thick, and so we can image very large tissue samples. Of course, what we would really like, going back to connectivity, are labels that get us closer to connectivity, and that's one of the things we're working very hard on. So for instance, myelin labels, or something else that will label axons and dendrites, but small molecules, so that we can get it in. Something that we really want. And of course, we're keenly aware that once we are capable of getting this data, then the big data analysis, something I believe you've learned about yesterday, will become a really significant aspect of this as well. And so with thanking all the people in my lab, most certainly Frank and Robert and Arco that have done a lot of the MRI, and Sven and also Anna Schutt that have done a lot of the clearing and the light sheet microscopy, and our collaborator Ralph Beluske, I want to thank you for your attention.